learners, it's Em from Sono Nerds, and this video is going to be on the male pelvis, looking at scrotum physiology. So why do we need to know physiology? Well, the testicular physiology is related to pathology at the cellular level. So we're going to talk about some microscopic anatomy, talk about what that microscopic anatomy does, and that'll later inform why some cancers develop and where they develop from. The other thing we're going to do in this physiology talk is talk about how sperm exits the body. And when we do that, we'll talk about a few more internal anatomy pieces. And then this will all relate back to other diseases that might affect the male reproductive tract. The testicles are both an endocrine and exocrine organ. So as a reminder, endocrine means that there is a hormone released directly into the bloodstream, where exocrine refers to the delivery of substances through a duct. There are two major groups of cells that we need to know that relate back to testicular function. And the first one are the Leydig cells. The Leydig cells are responsible for secreting testosterone. The Leydig cells are found out in the interstitial tissue, so outside of the seminiferous tubules, and they're going to respond to luteinizing hormone, or LH, which is secreted by the pituitary gland. So as LH is released into the blood, the Leydig cells will receive that message and start to secrete testosterone. Now this usually starts around the age of 12 during puberty. As the testicles secrete testosterone, it'll cause the primary sex organs to grow. In fact, the testicles themselves will more than quadruple in size, typically going from about a 1 to 3 milliliter volume to 12 to 25 milliliters in the adult male. Testosterone is a hormone known as an androgen, and in males, it'll cause the voice to deepen, it causes more red blood cells to be produced, it also causes bone growth and other secondary sex characteristics like the increase of muscle mass and facial hair growth. The other subset of cells that we need to know are the Sertoli cells, and they're going to receive follicle-stimulating hormone, or FSH. And FSH is also produced by the pituitary gland. The Sertoli cells are found inside the seminiferous tubules, and they are going to sit right adjacent to the spermatogonium. Now, the spermatogonium are kind of like the germ cells. They are going to be the cells that actually divide and produce the spermatozoa or the gametes. But the Sertoli cells are going to kind of act as this regulator, telling the spermatogonium when it's time to divide. And they know that by receiving the follicle-stimulating hormone. So we see that a combination of testosterone production and FSH are required for spermatogenesis. Again, as a brief overview and a very simplified overview, we have the hypothalamus, an area in the brain that releases gonad-releasing hormone, and that's going to interact with the pituitary gland. The pituitary gland, when it gets GnRH, is going to release both LH, luteinizing hormone, and FSH, follicle-stimulating hormone. The LH is going to interact with the Leydig cells, and that's going to tell them to produce testosterone. That testosterone is then going to enter into the blood of the seminiferous tubule. So that's ingredient number 1. As FSH is released, that's going to interact with the Sertoli cells. And the Sertoli cells plus the FSH plus the testosterone are going to talk to the spermatogonium, the cells that are going to divide to create the spermatozoa. So testosterone and FSH are required for spermatogenesis, or the creation of sperm. So it is inside the seminiferous tubules where spermatogenesis occurs. Again, as an overview, we've got the Leydig cells, which are going to produce testosterone in response to luteinizing hormone, which is produced by the pituitary gland. The Sertoli cells are inside the tubules, and they will receive FSH, also from the pituitary gland. The combination of FSH and testosterone tells the body to start sperm production. The spermatogonium, which are also stem cells in the outer wall of the seminiferous tubules, begin to divide, and these are going to create spermatocytes. The spermatocytes then are pushed toward the center of the seminiferous tubule, where they develop into spermatotids. Once they develop tails. They are pushed into the lumen of the tubule. And from here is where they begin their journey through the tubular system. We are out at the seminiferous tubules. The spermatotids have been pushed into the lumen, and they are going to travel through the seminiferous tubule into the straight tubules and then into the reedy testis. After the reedy testis, it'll go through the efferent ducts into the head of the epididymis. And as the sperm are pushed through the tubule system, they're going to be developing into mature gametes. Once they're in the epididymis, they do travel through the epididymis into the ductus epididymis, where they will travel down to the tail of the epididymis, awaiting their chance to exit the body. When it is time, they will exit the tail of the epididymis into the vas deferens. Once we are in the vas deferens, they will travel up the vas deferens, which enters into the abdominal cavity, travels lateral to the bladder, and then curves around to the ampulla of the deferens. The ampulla of the deferens is a little bit larger area, and that's going to connect in with the seminal vesicles. The seminal vesicles are going to produce fructose, a fluid that gets added to the sperm. The sperm and that fructose are going to go through the ejaculatory duct that is within the prostate, and the prostate is going to add its own prostatic fluid to the sperm, and at this point we now have semen. So we need fluid from the seminal vesicles and fluid from the prostate added to the sperm to create semen. The ejaculatory duct is going to join up with the urethra at the verumontanum, and at this point the semen is expelled out of the body through the urethra and the end of the penis. So let's just go ahead and cover that again with words. So we start with spermatozoa made in the seminiferous tubules. Those are going to exit the tubules through the reedy testi. They enter into the epididymis through the efferent ductules. They then reside in the head of the epididymis and really the whole epididymis to mature more. They're going to travel through the ductus epididymis, where they kind of wait and become dormant until it is time to exit the testicle. When it is time, they are going to exit the epididymis through the vas deferens. They're going to pass through the seminal vesicles where fluid is added and exit into the prostate through the ejaculatory duct. More fluid is going to be added by the prostate, and now we have semen. At this point, the ejaculatory ducts are going to enter the urethra at the verumontanum, and then they exit the body through the urethra. So go ahead and pause here if you need to write any of this down. So here's a brief description of each of those anatomical structures. Again, the vas deferens is the tube that travels superior through the inguinal canal, through the spermatic cord, and lateral to the bladder. It's going to end at the ampulla of the deferens at the seminal vesicles. The seminal vesicles then add fructose to give the sperm energy. That fructose and the sperm are going to go through the ejaculatory duct in the prostate, and the prostate is an organ that adds like citric acid, calcium, and other proteins to the sperm, now making it semen. At the verumontanum is where the ejaculatory duct and the prostatic urethra connect in the prostate, and then the urethra continues on as a tube that will bring semen out of the body and also doubles as an exit for urine.